All right. Well, welcome everyone to our discussion today. I'm Eric Wamwell. I'm an instructor, professor over at UMKC School of Pharmacy, and today we are going to be talking a little bit about alterations to the GI microbiome. It's something that is an emerging area of interest. We hear a lot about microbiome, GI flora, the dysfunction, causes that can be related to immune diseases, cardiovascular diseases, infectious diseases, and so hopefully today we can start to shed some light on what this topic is about. Today I've got DJ Flynn, who's a student at the pharmacy school with us, as well as Francis Kenyon-Dewey, another student from UMKC, to help us learn a little bit more about alterations to the GI microbiome. So first, it might be beneficial for our audience to understand what is the microbiome. And so Francis, could you help us out with that a little bit?
Yeah, sure. Microbiome or microbiota is a term that is used to refer primarily to a group of microorganisms that are non-pathogenic, so they don't cause illnesses, and they reside mostly on the open, exposed surfaces of human, either maybe on the skin or maybe on the mucosal layer of the GI and also genitourinary tract. And 70%, more than 70% of microbiota occupy large intestine, so that's a pretty significant number. Another word that you hear out there referring to microbiome is like microbiome itself, normal flora, good bacteria, or even friendly bacteria, so to speak. So normal flora, or the composition of the microbiota is in the GI is about 66% anaerobic, with two major groups of those anaerobes being bacteroidetes and bacilli class of firmicutes. According to a recent study involving multiple subjects, it has been suggested that human microbiota comprises of more than 35,000 species of microbiome, and that composition is not similar along the GI, because as the study has shown that in the upper GI, especially the stomach, going on to the intestines, going to the large colon, the concentration varies and the composition is also different, with as much as 10 to the power 14 in the colon, and only maybe 100 per gram in the upper GI. The other difference that also exists is across the GI lumen. We have different composition on the lumen that is not the same as the composition in the epithelial cells and also in the mucus. So there is a quite different number of species or different composition in different sections.
Excellent. Wow. So we've got a lot of pathogens living in our GI tract. So there must be a function to that. DJ, can you tell us a little bit about what their function is in the GI tract?
Yeah, of course. We live in a symbiotic relationship with these bacteria. They help us out a lot with food digestion, especially energy harvest and calorie uptake of things like sugar, amino acids, fatty acids. They also help us in detoxification of foreign substances, production of bioactive compounds like bile acids, steroids, eicosanoids, and also they help us break down things that we would otherwise not be able to digest, like large fiber molecules.
Yeah, another added advantage of microbiome is in the field of immunomodulation. We have some benefits, both systemic and local. The studies have shown that they induce proliferation of CD4 T cells, also promote the development of lymphocyte, also regulates the dendritic cell count, cytokines, and activation of natural killer cells. A study was done that had shown that animals that did not have microflora had some structural and functional abnormalities in the spleen, in the lymph nodes, and also they had decreased number of lymphoid follicles. And what happened is, when they were given supplementation of normal flora, they were given a species called Bacteroides flagellis. Those pathophysiologists seemed to reverse. So that kind of is an evidence that it does help in the modulation of the immune system.
Wow. So both absorption, metabolism of our food, as well as playing a role in just normal function, organ, tissue growth, immune modulation as well. So pretty significant functions from that symbiotic relationship that we have. Excellent. Because they have such important functions, one of the things we hear about is a disruption. And what are some different things that can disrupt our normal microbiota?
One thing that I found a major role in disrupting or altering our microbiota is what we intake, our food, our diet. For instance, a study shown that mice fed with the Western diet, which is typically what Americans eat, it's high in fat, high in sugars, and they compared that to low-fat and low-sugar diet, and it showed a relative increase in the firmicutes and decrease in the bacteroidetes, the two major taxa that Francis mentioned. And what that does is it enriched the genes that enabled energy uptake and calorie uptake. So the more calories led to weight gain, obesity, things like insulin resistance, diabetes, etc. Another interesting fact as far as our diet too is that another study showed that in mice that consumed a fructose solution, a high fructose solution for 8 weeks compared to a group of mice that just ingested water, it showed a 27-fold increase in the endotoxin levels, which that's coupled with inflammation, cytokines, hepatic stenosis, insulin resistance, as compared to those that just drink water. So I think we've underestimated actually how much our diet can not only just affect our body, but one area of that is our gut flora.
Yeah. To add to your point, another two factors or two areas that can also cause disruption of microbiota is invasion by enteric pathogen, because we are always exposed to the pathogen, from the environment. If there's any ingestion of those pathogens, and also maybe through the skin where we also have normal microbiota, if there is interruption through ingestion, we can have increased inflammation response due to that infection, which what that does is decrease the viability of the microbiota, which leads to colonization of the invading pathogen. So through that, we have decreased microbiome as compared to what it would under normal circumstances. Another thing that sometimes we might take for granted is something like diarrhea, which can induce disturbance of the homeostasis in the GI that leads to alteration in microbiota composition. It's also called dysbiosis. So those are other two additional things that can disrupt the microbiome.
One thing that's probably more applicable to us as pharmacist students or pharmacists is the use of antimicrobials and antibiotics. And it obviously depends on the spectrum of the antibiotic, whether it covers gram-negative pathogens, gram-positive, and anaerobes. But they alter the levels and diversity of our normal flora. For instance, a specific study by Perez-Coba showed that a patient that took 14 days of beta-lactam therapy showed drastic changes in the total diversity of the number of taxa of bacteria and the total number of bacteria. It hit its lowest point from days 11 to 14, but after the discontinuation of the antibiotics, in general, the regular flora that was there before the initiation of antibiotics grew back to its normal. But what they did notice is that after the treatment of antibiotics, the bacteria that were in there originally, that were disrupted, led to they were more prone to acquiring foreign DNA, which can lead to antibiotic resistance. And also there was pretty significant alterations in some of the metabolism that I mentioned earlier, with vitamin and mineral uptake, production of hormones, bile acids, and things like that. So even though in general the bacteria grew back, there was some alterations in there after the treatment of antibiotics.
So a variety of things really having a major impact on our microbiome, all the way from the foods we ingest to infections that we get to just mechanical diarrhea having an effect to antimicrobials also playing a role as well. So a variety of different things there. And just in general, are there any differences from, say, one person to another outside of these other disruptions that occur?
Yeah, in general, people from babies to adults or even different regions in the world, what I've seen is that there are significant differences. For instance, when a baby is in utero, they're considered sterile until they are exposed to the flora of whether they're delivered vaginally, the vaginal flora, or C-section, they're exposed to skin flora. But as they grow to about 2 years old is when they are considered to have the normal flora of an adult. But from the age of 0 to 2 years old, the major changes that we see is when they start going from like breast milk to solid foods, is when they start developing more of the anaerobic bacteria, acquiring that. Also, unique differences, like people in Japan, it's shown that they have a unique enzyme-producing bacteria in their flora that helps them with the digestion of seaweed, which is pretty prevalent in their diet. And also, kids in Africa have been shown to be more rich in the bacteroides, as opposed to the firmicutes, which what that leads to is better digestion of the high-fiber diet that they intake. So, depending on where you live and the diet, there's pretty significant changes and differences in the bacteria.
I guess that's one of the things we might expect, but it's interesting to hear the data on that from different regions, having different diets that you pick up the different microflora from. Speaking about the vaginal delivery, one of the things that occurs in vaginal delivery is there's usually some defecation that occurs during vaginal delivery, and so the infant, comes in contact with that as well and picks up some of that flora as well from the mother. So really interesting stuff on how that microflora develops from one person to another. Now, in these differences from one person to another, or disruptions, how are we seeing that impact disease progression or development?
From what I've seen, with the disruptions that we see, one of the major components is from this development of this chronic low-grade inflammation state. It's especially more prevalent in those that ingest a Western diet, so the high fats, high sugars, because they have more of the firmicutes as opposed to the Bacteroides taxa. What it does is the gut bacteria can initiate an inflammatory state, which can lead to obesity, insulin resistance through a substance called lipopolysaccharide, or LPS, which is an endotoxin that's found on the component of gram-negative cell wall. And what this can lead to also is decreased tight junctions in the GI tract, in which the lipopolysaccharides is able to go into the bloodstream in higher concentrations and induce these inflammatory response just throughout the whole body, which can lead to cardiovascular disease, can lead to inflammatory disorders like IBD, IBS, Crohn's, that those gut diseases, cancer, autoimmune diseases, allergies, even things like fibromyalgia are linked. So there's significant implications on the disruption. A couple specific examples is with obesity and metabolic disorders, depending on your microbiota, like I mentioned earlier, you could have different levels of energy uptake. So you're absorbing more calories and more energy, so that could lead to obesity, insulin resistance. And interesting, unique study that I found showed that certain autoimmune diseases like rheumatoid arthritis and multiple sclerosis in germ-free mice, the phenotype or the symptoms were absent until they recolonized those mice with certain taxa, and when they recolonize them, then the phenotype or the expression of these diseases came back into play. So I thought it was very interesting.
Yeah, certainly. So, I mean, from the variety of diseases that you point out there, I think it continues to give even more power to the impact of maintaining a good microbiome. So, what about, did you guys find any research on ways to prevent disruption to the microbiome? Anything that you ran across that way, Francis?
Yeah, one area that we found is the use of probiotics in prevention of antibiotic-associated diarrhea, which, like DJ said, that's one of the ways that we can disrupt the normal flora and then leads to diarrhea. So there has been evidence that's shown that use of probiotic in almost all the either oral or just parenteral antibiotics can lead to at least decreased frequency of diarrhea. So that's one of the preventive ways where probiotic use can provide some benefit.
Okay, so co-administering a probiotic with, or probiotic with an antibiotic to help prevent that initial disruption.
Yes.
One of the things that we've seen. Excellent. Now, what about if a disease state has already developed, is already in play? Are there things that we can do to help restore the normal flora, to help reduce symptoms?
Yeah, according to what I've seen, this is the area that it's still pretty much under study, but there are some diseases and illnesses that we have seen the use of probiotics as being efficacious. And I looked at a study that was done in Sweden, and they were looking at atopic dermatitis and allergic rhinitis. And what they did is that they took 60 children between the ages of 2 and 14 who had atopic eczema and dermatitis. And what they did with these children is they divided them into two different groups. And in one group they gave them, they administered probiotics. The other one was just placebo. And after 8 weeks, this was done for about 8 weeks. And after 8 weeks, they did find the benefits in the group that had probiotics. And what they did see was that the atopic dermatitis scoring tool, which indicates the presence of the dermatitis, was lower in the group that had probiotic as compared to the group that did not have the placebo group. Also, in this study, they also found that eosinophil protein levels were also significantly reduced in treatment group at week 8 compared to week 4, which was also another evidence to show that the group that had been treated with probiotics, which in this case is improving the function of the normal flora, had some benefit compared to the group that didn't have.
Yeah, it's amazing, like a reduction in inflammatory response as well as an alteration in the immune response of the patient as well, just through giving probiotics. Okay, wonderful.
Another thing that I've seen with, as far as management is more of a non-conventional, something that we might not think of, is through the use of fecal transplantation. So taking the normal flora of the GI tract of a healthy individual and implanting it in someone that has a disruption. Evidence has shown in certain things and certain diseases, like with C. diff infections, that implanting or a fecal transplant can restore the normal flora of a person just by implanting what should be there. It suppresses the bad commensal bacteria that have invaded. But then again, there's also some evidence that's inconclusive. When they do a fecal transplant, in some cases, it's shown that the good bacteria that's been transplanted can acquire resistance, or that the flora, the bad bacteria that's in there, can suppress the good bacteria that comes in. So it's kind of an emerging topic, and more studies are currently being done on it. But yeah, I think we'll have some good emerging evidence on that.
Yeah, another area that we've seen the use of probiotics being helpful is in the case of H. pylori infection, which is very prevalent in the population. It leads to gastritis and inflammation of the stomach, which can lead to increased acid production and maybe consecutively leads to stomach cancer. This illness or this infection does not have cure currently, or there's no vaccine for it, and this is an area where probiotic therapy has been proven to come in handy. It's been shown to decrease the growth of the number of H. pylori bacteria in the group that is treated with probiotic. Studies have shown that administration of one or two combination of probiotics together with the current treatment for H. pylori help minimize antibiotic resistance and side effects from the treatment of H. pylori. So that's a very beneficial area. Yeah.
Wonderful. So, some ways we can help restore the microbiome once there's disruptions, although not perfect. Some evidence to say in populations we can have some benefit. Interesting. Wonderful. So, definitely an area of evolving research. Leads to a lot of tangential questions about how these pathogens can really have these overall effects on our normal functioning, on disease states that affect us, and probably all kinds of emerging areas of research underneath that umbrella. DJ, what are some different places where we see some of that research being done?
Well, they're trying to see what is considered a normal, or see if there's like a universal flora that would be good for everyone necessarily. Also, like I mentioned with the fecal transplant, they're looking to see if it can cure or at least decrease the symptoms in several different diseases, more so the autoimmune diseases and inflammatory diseases. So those are areas as well. They also raise questions, for instance, like wondering if there's like a difference in the morning and night. There's like a different morning flora or a night flora, or just if our regular flora has different activities during the day versus during the night, depending on hormone levels and things like that. Also, with like Francis mentioned, with diarrhea, we also wonder if how laxatives might affect our flora, or even constipation, like opiates or whatever substance that causes those kind of things, how they can in turn disrupt our flora. Another question that has been risen is, how does cohabitation affect our normal flora? If it does it affect the man or the woman more? There's just lots of unanswered questions that in this emerging topic that there's tons of area of research to be done.
Definitely. Well, this is all
cool.
been very fascinating. Thank you guys for your work on this. We've got a number of people on phone call joining us, and just take a minute to open up to any questions that they might have for us at this time. All right, then. Hearing none, I think we'll just kind of leave it at that today. Certainly, different ways that this is going to apply to a variety of different practices, whether you're working in allergies, whether you're working in infections, immune diseases, GI diseases, obesity, cardiovascular disease, diabetes. This GI alteration can have an impact on a variety of these different areas, and so certainly something that we should all be exploring, looking into, and having a better understanding of the effect in those different populations. So again, appreciate your time, DJ and Francis. Appreciate your time for joining us, and we'll call it a day. Thank you.
Thank you.
Thank you.